This is a panel session about the most promising intervention strategies in aging. By intervention strategies, we don't mean intervention, pet intervention, because those, I have them already. So in the first night, the night yesterday evening, I've circulated a little piece of paper, so we've had a beer before, so that may excuse things. And so I know that some of them think that, for example, just testing in mice is stupid, sorry. And another one thinks that we should make some genetic engineering. So it doesn't say, but maybe that's in people. So we see that we have various views. Everyone has a specific pet intervention. And so here we're going to discuss about strategies. And so the question I would like to ask first is when you have a strategy, so a strategy for interventions, the question to ask is what for? So where to test it, if it's in mice, in people, for research, for application, for everyday life. So that's my first question to everyone.
You've got the microphone, Sven. You go first. You first.
I will say we have, you told me, especially the industry, the insurance industry has large databases where they keep track of people and what factors they have. I don't know in what detail those databases are, but we should have studies where we look at the use of drugs, the use of ideally we will have large studies across the population where we look at the use of drugs. We might want to take some blood samples, do some things like that, and then look for factors. So that's one way to go for it. Another way is, of course, the mice and other animal research. Personally, I think using C. elegans and Drosophila really has shown its limitations, and I'm more in favor of some of the more rodent models. And also some of the more new models, such as naked mole rat, for example, to use, although their long lifespan doesn't really allow very easy intervention studies to be done on them.
Well, I kind of agree with Sven. I'm a big fan of in silico computer-assisted models, where we would be able to model human response to a variety of drugs, because right now there are huge databases available on both genomic, transcriptomic, metabolomic, proteomic level, so all the omics data is out there. It's like Swiss cheese still, so there are lots of gaps. For example, there's more gene expression data, there is much less metabolomic or genetic data. But there is enough data available for humans to be able to tap into that data and find if we can mine some interventions from this data. So that's what we're kind of trying to do. But really what we need to do in order to have interventions with a solid set of biomarkers of aging. Well, something that we can measure in healthy patients and something that we can provide to large pharma in order to create business models around some of their products. So they would be able to test their products in healthy patients, see if their products change a set of biomarkers, and if they do, they would be able to address some of the regulators and get those products on the market as prevention.
Okay, I will just make the question slightly precise to resume and evolve. So from Sven, I hear statistics. The statistics in humans and in mice might be exactly this. From you, I see more personalized statistics. So right now we are still in the statistics, well, in terms of applications or in terms of how to find the results, right?
I agree, so statistics, right?
Okay, so let's stay at such a very high level, just to start having an overview.
Yeah, I was tipsy when I wrote that, and I said that research in mice is stupid. Obviously, I don't mean to be offensive. I think there's an inherent contradiction in asking how do you test anti-aging technologies, should you test them in mice or in silico? Because how are you even supposed to know that something is a candidate without having the information in the first place? And I agree with both what's been said so far about relying on databases and in silico medicine to, in the first place, know how you're supposed to approach it. And even though these databases, I mean, you said they're huge, I think they can definitely be even huger. And with regards to comparing sort of results obtained on mice versus humans, even amongst humans, there are differences when it comes to what drugs work best for different cancer patients. And so when you have those differences amongst humans themselves that have to be tested before you can optimize treatment for each person, when you look back to mice, you realize that actually the resolution is much, much lower. And I think as proof of principle, it may be justifiable to do some elegant experiments in mice just to be able to say, yes, you can do this in humans. But when it comes to really complicated, very sensitive quantitative experiments, it becomes more difficult to actually be confident about the data in the first place, and then you end up having to test it on humans anyway. And of course, you do get some negative side effects when you get to that point, where you might as well have just gone straight to it in the first place, and the same damage would have been done nonetheless.
Okay, so I agree with most of what you said before. The only thing I would add, so first I guess, as you pointed out, it depends on what you're testing or what you know already about what you're going to be testing. I guess I wouldn't say mice. I would say rodents have limitations. One of them, the fact that a lot of there's a lot of strain-specific effects. So ideally, you do experiments like the ITP do in more genetically heterogeneous strains. If you're trying to look for lifespan as an endpoint, you cannot do it in humans. So you have to use short-lived model systems, of which rodents would be better than C. elegans, at least in relevance to humans. It also depends a little bit how big of an effect you expect. If you expect this to be a rejuvenating therapy, then you don't actually need biomarkers. It's going to be so fantastic that as soon as you put people on it, it's going to have an effect. So it also depends on what the expected outcome is going to be, and based on that you decide on particular endpoints, and then you decide on what exactly you're going to test.
Yeah, I think the main thing I would add to what's been said so far is that we really absolutely need to focus on late onset interventions. We need to do testing on older animals. And I think if we do that, then a couple of things become simpler, in addition to the obvious fact that we eventually want things that will work on older people. First thing is that the personalized aspect becomes less. And the reason it becomes less is the second one, which is that the therapies that one will be testing will typically be rejuvenation therapies. They will be ones which actually restore some aspect of the physiology of the animal to a more useful state. And that in turn means that one doesn't necessarily need even to look at lifespan. One can look at the relevant aspect of the physiology, whether it's immune function or grip strength or whatever it might be. So I would say this, the standard mouse models, I agree with what was said about not using an inbred strain, but the well-studied mouse models are still, it seems to me, the place in which most of the actual productive legwork is going to be done for the next few years at least. I think that is borne out by one thing I mentioned yesterday, the fact that even very preliminary results in mice, if they show bona fide functional rejuvenation in older animals, are very interesting now, even to the commercial sector, which certainly is an essential step in the eventual delivery of drugs that really work for you and me.
Okay, so just to resume, we've seen that there are many ways we can go from what we know or what we believe we know. We can have statistics in humans, in mice. We can base things on genetic analysis, and we can test bigger things in mice. So now I'd like to change a little the axis just to dig the space with time. So here we're focusing about the most promising intervention strategies in aging. What do we think we can achieve the best, the most promising intervention strategies along with time? Just to give an example, one may think that gene therapy is going to have big effects because genes are the way the body functions to some large extent. And we need to play with the right buttons on the genes, but perhaps it's not today for humans. So in terms of scale in time, what do you think are the most interesting strategies for interventions?
Well, a couple of years ago, I would probably have said that the short-term, near-term things that we need to be looking at are pretty much the same things we have been looking at, geroprotective drugs and so on, simply because nothing really useful is going to come along. But increasingly I'm thinking that, at least in terms of demonstrating bona fide rejuvenation in model organisms, and perhaps even in the clinic, we may be only a few years away from doing things that looked much further away not long ago. I mean, of course, we all know about CRISPR, the advances that have been made lately in gene editing, in improving the safety and efficacy of manipulation of the genome in vivo as well as in the dish. And I believe that that's really completely changed the landscape for the time frame of a lot of the interventions that we're going to need if we want to make a real difference to the ill health of old age.
Thanks. So I agree in the sense that, you know, what we have, I mean, geroprotective drugs, I mean, they've been covered extensively throughout yesterday and today. The effects are relatively limited. You know, metformin is like 5.8%, even rapamycin like 14% increase in life. So in mice, which God knows how much it's going to be in humans. So sure, they can have applications, but they're going to be fairly limited. I guess it also depends how ambitious you want to be. I think you're fairly ambitious, aren't you, already in that sense? But I personally think that the field should be more ambitious than that. And in that sense, you have to think about really manipulating genes. Now, gene therapy has been fairly disappointing thus far. CRISPR, I guess it's emerging technology. Fingers crossed it's going to work, but again, there's still a lot of work to do in implementing those technologies for human therapies. So hopefully, my hope at least is that the field more ambitious and more striking interventions are developed based on these emerging technologies.
Yeah, I think CRISPR-Cas is really, really interesting, and you're essentially permanently replacing a bit of your genome that can be passed down indefinitely, and that's as direct a form of editing the information upon which your body is based on as it gets. And I think that ties in really well with the information technology that is required to manage that sort of technique in the first place. But I still think there's far more information that is required, even about the genome itself, before that can be implemented well. And I think once that information is gathered en masse, then that will become much easier. I also think that connecting that to existing computer interfaces that can make it easier, in the sense that instead of having to sort of inject something into your body or somehow force it into your cells, if you have a communication interface with your body, then that can be controlled remotely, and I think that can make the whole difference.
I agree with the previous speakers that gene therapy probably has the highest promise at the moment, but I think the roadmap for interventions is very plastic, and we're going to see a lot of areas converging and resulting in additional years of lifespan. And I'm not only talking about geroprotectors or gene therapy or regenerative medicine, but also self-driving cars and minimizing risk. So advances in information technology, advances in diagnostics, advances in monitoring and prevention of the disease, that's going to help us live longer and achieve, and live to the point where we would be able to intervene intelligently into our systems. But what's interesting is that I believe that some of the geroprotectors are already available today, and we just need to repurpose some of the already available drugs. There are tens of thousands of them. Some of them went through clinical trials. We just need to retest them, maybe first in silico and later in vivo, for their efficacy in terms of extending lifespan and suppressing the age-related processes, pathological age-related processes, and perhaps repairing some of the damage. And one of the very interesting outcomes of the event last week in Basel was that Novartis took one of their aging drugs, well, potential geroprotectors, everolimus, it's a Rapalog, into clinical trials, well, not clinical trials, it's internal clinical trials, where they're administering Rapalogs, everolimus, to patients as a vaccine potentiating agent. So essentially giving elderly people everolimus for several weeks before inoculating them with a vaccine. So, and that's one of the strategies for getting a geroprotector on the market. So hopefully we'll see some results from that study, and that's a very ambitious study. And hopefully that will give us a little bit more data about how some of the geroprotectors work and how to get them on the market. So again, promises with gene therapy, but currently I think we should also explore what's available right now, because I wouldn't put a gene in myself before I get it tested on other humans at least, and that would take a long time.
So the previous speakers are almost all mentioning the CRISPR system to permanently change a piece of DNA in us. And the problem that I haven't heard any of you raise is essentially when it is not for a very specific disease, how are you all even going to get this done, or have clinical trials or any other thing going on in humans? You know, it is regulatory very difficult to do. To get approval for such a study will be really difficult. So that's a real limitation that is probably going to hold back this technology a little bit.
I don't think it is. Really much better.
So this comes back to something else I said yesterday. The medical industry find ways to get things approved when they think they can make money out of them. If something needs to be for a specific disease in order to get into a clinical trial or to get approved, then it will be declared to be for a specific disease, whatever disease happens to fit the therapy best, even if the overall intention is actually something more general.
Well, I hope it works out, but you know, it's not easy to have something declared the disease. I mean, there has been, you know...
No, you don't do that. You just say that the therapy is for the thing that's already been declared a disease.
And none of label use, right?
Yeah.
If that works, if it works, we will see. We will see. But probably, I guess there is the option more of developing things like cross-link breakers, like specific therapies that deal with certain types of drugs. The Science Foundation has, for example, these devices, if you can call it like that, where you basically can remove senescent cells from the blood using magnetic beads, which is obviously easier as you are not intervening in the genome or anything that has potentially lasting effects. So that's probably going to be things that you can much quicker and easier bring into applications or tests.
So if I want to challenge things a little, so do you have a view so I will ask 3 questions to everyone that will be easier. So perhaps give a date of when we would have a genetic intervention performed in humans that is really for longevity, and what intervention that would be. That's the first question, so very specific. And of course, I'm sure you will argue. And the second question will be so I'm going back to the PET results. The PET drug that you would imagine soon, what would it be? So this is no need of date, that's soon. And then the non-genetic, non-drug intervention that you think is most promising, except out of this. So it's a bit a complex question. It's 3 questions, but it's in order to have a bit of things to discuss.
Okay, I'm being given the hard task of coming up with a date when a genetic intervention specifically against aging will be tested in humans, which is really hard to put a date on. I honestly have a very difficult period putting any number on that. Let's say maybe in the next 20 years, specifically against aging. That is not like, when you use against some, you say it's a disease, and so, I mean, 20 years I think is optimistic. You will have to change, the whole concept that we can also treat aging itself, right? That is a target for intervention. So, I mean, 20 years I think is an optimistic thing.
I said for longevity, to live longer, yes.
Yes, well, that's, to extend lifespan, yes. What will that be? What target? Well, you of course have targets within the insulin IGF-1 pathway, which are the classical ones, which have most of the evidence behind them. So, which will be an obvious one to go after. You have FOXO3A, which already has been shown in human supercentenarians to be increased. So, I mean, that will obviously be one of the obvious ones to go after, as there is the best evidence for it. Oh, yes, I need to do the other questions.
The second question was soon, very soon. What is the big promising pet intervention in terms of drug that you would see come for real?
Well, I mean, I see that a lot of people, myself included, are already taking metformin, so I guess this is already breaking through.
The last question was, except from genetics and drugs, what is a very key intervention?
A key intervention? Well, I guess I already mentioned the device to remove senescent cells. That will be one thing that is actually already done in mice, so it seems to work. I don't know to what extent it has been shown to have beneficial effects. Aubrey can certainly expand on that.
Thank you. I agree that probably the gene therapy that's going to be targeting longevity, that's within the next 20 years, maybe sooner, hopefully sooner. If I were to select a target that I would try, I would look at myostatins and maybe the ACT2 pathway, so things that lead to, well, anti-sarcopenia vaccines that also lead to, the Belgian blue phenotype. You've seen the Belgian blue phenotype, right? Oh, and bulls, it's bulls without fat and lots of muscle. So if I were to intervene, I would really like to start from there. As a matter of fact, some of the large pharma do have some of the myostatin inhibitors and ACT2 receptor inhibitors as well on the whole pathway. So that's something that I would experiment with, and I think that once bodybuilders get a hold of that, and we see some longevity effects, that might be sooner than later. In terms of the major intervention of choice that is not a drug at the same time and not a gene therapy, how many of you are aware of the deuterated, isotopically fortified organic compounds? That's the work of Misha Shipinov, which has been commercialized in a company called Retrotope. So what they do is essentially they take an organic compound that gets oxidized really quickly and leads to deleterious effects. So like, for example, lysine, or you take linoleic acid. And you can fortify the double bond in some of the polyunsaturated fatty acids just by substituting one of the hydrogens on one of the ends of the double bond with deuterium. And it's much more difficult to get into the transconfiguration or break the bond. And that prolongs the life of, well, quote unquote, prolongs the life of cellular membrane. So it extends the time the membrane remains permeable. And some of those drugs are already being tested in Friedreich's ataxia patients, and in mouse models they're tested in Parkinson's patients. And if that drug would have been available in the markets, I'm talking about the polyunsaturated fatty acid fortified with deuterium, just one isotopic substitution, I would be taking it right now. And I think there is a lot of promise in those drugs. I know there is a lot of skepticism in the area, in the field, but large pharma is interested. They just got a very large amount of funding, and I've seen the results in animals. And by the way, I have a conflict of interest. I was an early investor in this company. But I invested just because I believe in the technology, and if it would have been available, I would be taking it right now. So I think that would be a killer intervention. Instead of scavenging reactive oxygen species, you would be protecting the oxidized organic compounds.
May I ask, I think you provided it before, you said regenerative medicine with organs, I think you said. So, or maybe if you want to give one example of non-genetic, non-drug intervention.
Well, that would be regenerative medicine, yes. So maybe induced pluripotent stem cells gaining therapeutic potential.
Do I have to still give a date?
Yes, that's the most interesting question.
What is the date? What is the date for?
So, good question. The date is for when people, not necessarily many, will take a gene therapy in the goal to live longer and healthy.
So specifically gene therapy?
Yes.
I don't think it would be marketed as the gene that makes you live longer. But if it were just something quite that people could view as something every day, that maintains their body rather than sort of makes them live longer, because obviously it couldn't guarantee that itself. People sort of already do things in the way of that. But for the date, for the sake of the date, does it have to be just one person, or just how many?
Let's say a few persons, so let's say 5 minimum.
Well, presumably they would be people in some sort of a trial then, or
Let's
crazy
say
people
yes.
in a garage doing their own lab.
Well, okay, fine. Yes.
Maybe
Okay.
they've already done it.
That's clear.
Maybe they've already done it. So minus 2.5 months ago.
Okay. And what about the drug?
What drug?
So if soon there was a drug that would make a big impact?
No, I don't think so. I think just the word drug itself is too focused on disease, and obviously aging isn't a disease, so I don't see how a drug could really make a difference. I mean, it could make a big difference relative to our current life expectancy, which obviously would be a big deal, but in the big scheme of things, I don't think that there will be the drug that people will start popping like Skittles, and it'll just be, living ever. I do think it's more of a maintenance thing, like brushing your teeth twice a day. I mean, you don't really think this disease, you have bacteria growing rampantly in your mouth all the time, and fungi on your tongue. You just brush your teeth twice a day, you don't think about it, and then you have a nice feeling mouth. It's not something, it's not so disease focused, it is about every day. As the previous talk said, it's about the definition of health and well-being, so I wouldn't see it as a drug. It's just merely feeling okay and doing something to prevent an imminent disease, like if you find out that you're going to get cancer, you're going to do something about it. But when it comes to day-to-day feeling healthy, it shouldn't really be seen as a drug, because then your everyday state would be a disease that you're trying to prevent or treat. So I wouldn't say a drug.
Okay. So like a toothbrush?
Like a toothbrush, yes. A toothbrush for your extracellular matrix.
and about an intervention that would not be.
I'm also a fan of stem cell potential therapies because the idea is that your body degrades, and so if you know a lot of the reasons behind deterioration in the first place is because evolution didn't care, and potentially because there was a severe energy constraint. But I think now that we have so much energy capacity that we've left outside of our body for computers, for building skyscrapers, etc., we have a lot of access to energy. We haven't incorporated it into our bodies, and we still live in these bodies that are energy constrained. And I think that's obviously kind of ironic, really, that we create things that last forever, and we can do so many things. But yeah, we left our bodies in the dark age, even though we have the energy now to fight back to what evolution can give us, because there was a problem with energy then. We can now make this energy. It's just a matter of connecting it to the body, and then it shouldn't be limited anymore. So from that point of view, making cells, making tissues, making bodies, because we have the energy to do it, we don't need to eat leaves or something to get the energy, is obviously where the stem cell therapies can come into. And if you can replace damaged parts with new parts, then that's obviously easier than trying to fix the damaged parts, much like sort of buying a new car or whatever. Another thing is the interface between the genome and proteins, in the sense that, as was mentioned before in CRISPR-Cas, you make a change in the genome and then you expect to have a different protein circulating in your body. You can also have, obviously, the reverse of putting a protein in that changes something about the epigenome, for example, that creates a difference as well. And having sort of protein nanostructures interacting in the body in specific ways that obviously require a lot of information to be gathered in the first place, I think would also be a good intervention on a sort of mass scale.
So let me see if I remember all the questions. So the first one was the time, right? So when are we going to have a gene therapy intervention to extend lifespan? So regulatory factors aside, as you mentioned, it could be done in someone's garage. Yeah, I don't think it would be that long. 10 years maybe? 15 years? That would be my guess, but obviously it's guesswork. Which gene to target? There's a few genes that have been mentioned, IGF growth hormone pathway associated with aging, some genes associated with longevity. I do think for practical reasons you would go for a gene that would be expressed in specific tissues, just because, doing CRISPR, or gene therapy, it's difficult enough already in one tissue, so to do it in the whole body, I think that's beyond our current technology. So you go for one tissue, and so I would guess maybe the immune system. So you would have a gene therapy for some immune system related gene, hematopoietic stem cells maybe, or the thymus, that would rejuvenate the immune system, and that would decrease inflammation and improve the immune system to increase lifespan. So that would be the rationale behind it. Whether it works, I don't know, but I can see it more feasible than trying to change a whole bunch of tissues. What was the other question? Drugs. As I mentioned, most drugs have limited effects now. I don't really know which one. I personally would not take a drug to extend my lifespan. I think a number of people have made this point that there's no replacement for healthy lifestyle. So unless there was very, very strong evidence, I wouldn't take a drug, and I don't see any of the current drugs that we hear about to have enough evidence for that. And last one was no drugs and no gene therapy. Again, it depends on how much you want to achieve. I guess right now, and this has been mentioned in the conference already, you can just exercise. That's contributes to healthy aging and probably slightly extends lifespan, although its effects are going to be modest. If you're talking about regenerative medicine, sure, that could have the potential is much greater. But we don't really have therapies at the moment to test, but hopefully we will have down the line. I think that's it. Was there any other question?
No, thank you.
Yes, it's useful being last for a question like this. For gene therapy, if I were not going to cheat, I would say probably 10 or 15 years, and it would probably be a gene editing type therapy, probably using CRISPR or something like it. I think the most obvious thing that it would be to correct one's ApoE status, if one was ApoE4, so as to, hypothetically anyway, to postpone Alzheimer's and atherosclerosis. But I am actually going to cheat. I think it could actually be more like 6 or 8 years, because I think that the way it's going to be done is by ex vivo genetic modification of stem cells. In particular, I think I've got high hopes for a project that we are doing to address atherosclerosis specifically by incorporating a new enzyme into macrophages so that they can break down the oxidized cholesterol that currently is the major source of atherosclerosis. We've already got proof of principle in cell culture of this that was published 2 years ago now, and I think that there's a respectable chance that we'll get that going through mouse models of atherosclerosis within the next 3 to 5 years. After that, getting it into the body, I think the simplest way to do it is going to be by taking hematopoietic stem cells and modifying them, making sure we get the modification right, and then doing a bone marrow transplant, because I think that only a minority of one's macrophages are going to need to be augmented in this way in order to have the relevant therapeutic effect. In terms of the drug, I am broadly speaking pessimistic. I don't really think we have very many candidates left. The only thing I would mention relates to something that came up in a couple of talks yesterday to do with transthyretin amyloidosis in the heart, which has until recently been thought to be something that really only matters in very old age, but which last year was revealed to have an enormous effect on longevity in general, especially in terms of the ability of rare protective alleles to extend longevity. If anyone's interested, I can obviously find the citation somewhere, but the study basically showed that the one particular allele of transthyretin, which appears to act as a stabilizer of the tetrameric form and thereby an inhibitor of the amyloidogenic form, of the amyloidosis. And it turns out that there are already drugs that do exactly the same thing, and which are used for patients that have other mutations in transthyretin that accelerate amyloidosis. But it seems very much to me that those drugs would be likely to have the same effect as these protective alleles on normal people, and so that could be very exciting. Finally, in terms of the non-drug, I think I agree with Alex, actually. I think I would go for the deuterated fatty acids, which are extremely exciting. I've been following that work ever since its inception. I think Misha probably spoke to me about it before he spoke to anyone else about 10 years ago, and I'm delighted that it has gone forward. It's something that he published a couple of papers in Rejuvenation Research, and the company is now doing very well, so I can see that getting to the market really quite soon.
Okay, thank you. For me, I think we've done the time and asked the different questions. I'm just letting a few more seconds because due to those questions I have organized a sort of order. But if you ever want to discuss once more, please let me know, because it's fine. Okay. And I was just thinking, maybe in the audience we have someone from the pharmaceutical company or a medical doctor, just to have a view, because we've come to quite impressive changes. We were talking about gene therapy, about things that are quite surprising, and so I was wondering if in the audience we would have one person, either a doctor or a medical doctor, I mean, or a pharmaceutical company, just to give a little view, because we are talking about the future technology, and we have to confront with the realities outside of our world, as we've just said. No? Okay. Yes? Okay.
Just a comment on the potential of gene therapy. I completely agree with the comment of the last, this second last speaker, that is quite unlikely to be successful on the entire organism. So if you want to do a gene therapy to increase lifespan, I see it as very remote, not because we don't know enough about the genes, but because the problem that has made gene therapy very unsuccessful on solid tumors and solid organ is delivery. We have very poor technique of delivering genes efficiently to the cell in the organism outside the hematopoietic system. So I think I see it as a very big problem that still has to be solved, not only in aging research, but to use gene therapy practically.
Thank you. I think, in fact, I think we were to finish at 5. That's why I was stopping. But maybe we have time for a few questions, I guess, if we want.
I want to ask you. Can I ask a very precise question to the panel? And you just have to give a short answer to that. No explanation needed for that. If I can help you maintain one organ in your body for as long as you want, which one will be your choice? Remember, rest of the body will still be aging. Give me your 5 choices, or it may be same organ, I don't know.
So, Aubrey.
Yeah, no, I don't have an answer. There's no point in having one organ working.
I'm forcing you to give, please.
There's no point. What would be the point of having only one organ working?
It's bare.
Okay, so he doesn't want to commit on it.
I go with the brain.
Jaws and brain.
Yes.
Well, seeing as the brain goes last, I don't go for the brain. The skin is the biggest organ, but then that's superficial. So can I say my circulatory system? Does that count?
Okay, because I'm saving time also. We'll think about that later on. Why he? What about you, Alex?
I would go for the brain.
So can you say the question?
One organ which I can definitely guarantee for you to be maintained fully functional, non-aging forever, or as long as you want to live. But one organ. But rest of the body I cannot do anything about at the moment.
The brain.
Huh?
The brain as well.
Well, let's then think about it after when we have time.